So we left off on this beautiful dimer that we had formed, and next we ask, well, what difference does it make? For that, we go to melting curves. You can see the protein that it has an ALS causing variant melts at right around here, 45, and this is your body temperature, so this thing is melted, that is unfolded within your body. But if we add these crosslinkers, these maleimide crosslinkers, and then we give the protein back its metal, it stabilizes by some 40 degrees, and that's the most I've ever seen anything stabilize a protein. Not only that, it's inactive, the protein, but when you put back in the crosslinker and some copper and zinc, it regains activity. So this is inactive or less active, and this is much more active. So it's gaining activity as well. So that's cool. So now I'll take you on a little vignette of what we decided to do next. So we had these cysteine pairs, and we knew that this would work. We started thinking more generally, like, well, what about how many other cysteine pairs are there? Because this is cysteine 111 in SOD, but there's also some we know in DJ1. And so we came up with a type of compound called a cyclic disulfide, shown right here, that would take the place of these toxic maleimides and still be able to cross-link these cysteines. And the idea is that in a bunch of diseases, this is what happens. Two subunits come apart, just like that. There was a dimer and it becomes a monomer. Just like in an ALS and in Parkinson and A beta, there's all kinds of bad things that happen like that. So, and at the end of the lecture, I'm going to go into transtreatin with you, which also does this. Sometimes, though, it could be tau or progranulin, and a domain in a protein falls apart, and then the protein aggregates, and that's just an unfolding event. But in any case, we have these two cysteine pairs, and if we use a cyclic disulfide, it will, this disulfide will move over, hit the cysteine, break, and you'll have a thiol disulfide exchange. And then it'll go over here and another thiol disulfide exchange as long as you have an oxygen on it. So anyway, most important thing is we found some molecules to bridge these. So here's SOD1, we've seen that. Here's our cyclic disulfides. This is how they work. They start out as a cycle. They bind one over here, then they're bound here, and then they entropically favorably bind over here. Okay? It's reversible if it's a single cysteine, which is great because you don't want to bind single cysteines, a lot of important enzymes, but once it's bound to two cysteines, it's not reversible. All right, so we actually screened all crystal structures in the PDB, some 100,000 of them, and found all of the proteins that have these, by the way. So we have a bunch of targets. Here's something about targeting DJ1 cysteine pair. I have a similar thing on SOD1, but I guess you're probably tired of hearing about SOD1. DJ1 causes Parkinson, and just like SOD1, its dimer falls apart. And just like SOD1, there are these two cysteines here close to each other wanting to be bound by something. So here it is. Just take this rapid mass spec based screen. You have your control. Here's a monomer. These are deconvoluted, obviously. DJ1. Add this beautiful cyclic disulfide. You go from monomer to dimer. Okay? Very simple. We then went and got a bunch of these cyclic disulfides. Look at these guys. Tried these. Just within these boxes, these two, we tried these compounds. Did some structure activity. And this is the idea. Check this out. If you can find something that's in this region or this region, great. If you find everything here, well, nothing worked, right? Well, I've already sort of spoiled it and showed you that at least one thing worked. Most of our compounds didn't work well. A few of them were non-selective. And some of them were very selective just for particular proteins, which was great. Those are the structures if you're interested. And they enhanced stability, like you want to do. And then this is the coup de grace for us at this point. This is a monomer. Starting here, very low concentration, you have a monomer of SOD1, and as you get to about 5 micromolar in cells, the protein dimerizes. So this is pharmacodynamics-ish, but it's definitely target engagement, and you're watching our little druggy pre-drug stabilize SOD1. It does the same thing for DJ1 with a 5 micromolar EC50. And we've been improving these cyclic disulfides. And this one you probably heard of is lipoic acid, and this is one I can take myself. All right. Now, one of the interesting things is other groups were trying to do the same thing to bind the dimer. So look, this is dimer. So they went and did in silico screening, and they just looked around this pocket here, and they found a bunch of compounds that should go into the dimer, and they said, hey, it's stabilizing the protein. All of those compounds died because they didn't clear plasma. They made a second generation of these that did get through plasma, but when they crystallized them, dun dun dun, they were all binding right next to the tryptophan 32 I told you about earlier. So they missed the mark, but at least their compounds went somewhere. And that segues into another type of thing you can do with mass spec, and I'll just show you some of our data, but companies are doing this like crazy. You can screen 1,000,000 compounds in silico, see what binds the spot you want to bind, like that TRP32 pocket. There's one right there. You can buy the top 500 scoring compounds, and then look, this is a messy signal. It's FTMS, where you just throw in 50 compounds with the protein. Why not? You have the resolving power, and then you just let the proteins compete. So you have your SOD1, throw in a bunch of compounds, and say, hey, this compound is my best compound. It has the highest affinity because it has the highest signal. And in that way, we've found a bunch of different in silico hits for TRP32. That project's behind the cyclic disulfides, but we're going to keep going. And they do stabilize the protein. And that will be the end of the SOD1 story. Now I'm going to take you on only a snippet of an amazing story by Jeff Kelly's group out at Scripps. He has been studying transtreatin for his whole life, and it's the same story I just told you. You got to, except it's a tetramer and it destabilizes, and you want to stabilize it. So read this article, The Transtreatin Amyloidoses, from delineating the molecular mechanism of aggregation to a regulatory agency approved drug. The idea is if you have a tetramer with the ligand, it's going to uh, stabilize. Okay, and just like we talked about before, all of these nice conformational tools that we have were put to use in transtreatin, and they could see that, look, we have this native step, we have a partially unfolded step, and then it aggregates. We want to stop it either here or here, but we got to stop it. An important thing that they realized is that oligomers tend to go like this. 1 monomer, 2, 3, 4, and once they get up to this nucleus, we've talked about this previously, then all heck breaks loose. It's energetically favorable for that nucleus to aggregate, and it's a very bad thing. And they learned about how many proteins they needed for their nucleus for aggregation, and they want to avoid that. So they did some structure-based drug design. They got some things that would fit in there, and here they are. This is and they did a lot of medicinal chemistry. They checked how much they helped tetramer dissociation. I just looked the other night, and actually the EC50s of these for aggregation are similar to our compounds, where they're around 2 micromolar. But when you add these compounds, what's important is look at that. They could make the barrier for this transition state higher, make it much more unlikely, which is great. In other words, stabilize the protein. They then, once they had a few things that bound, they did structure activity-based medicinal chemistry optimization. So they added different moieties. They figured out where the best compounds now bound, and look, this one forms a covalent bond, which is kind of cool. And there it is, finally. This is tafamidis, excuse me, in its bound state, and it's been approved. They did some after the, I mean, I should say before it was approved, they had to do preclinical ADME-tox, put in a couple of animals, jumped through some hoops, went through phase 1, phase 2, and phase 3 clinical trials, and now it's approved in Europe, an academic lab that made a drug. Now I should tell you, and I have said it before, but transtreatin is fascinating. Transtreatin is in your blood, and it is believed by a lot of groups that sets the ultimate, today, it sets the clock on how long we can live. If we survive Alzheimer, cancer, all these things, we get to be 110, 120, this protein aggregates and kills you. That's just it. That right now is setting the upper limit of our lifespans. So with that, I hope you've enjoyed at least a little bit the process of going from structural discoveries to validating those with genetic tools and other tools to drug development, and in the case of Jeff Kelly stuff, to clinical trial and approval. Oh, and actually, this is funny. I just had a patent approved today. I didn't even think about that for the cyclic disulfides. Yay!